Today, let us learn about oogenesis, the formation of egg in human. Do you know the fact that in female, stages of oogenesis starts from the fetal stage? Yes, when the female fetus is just 25 weeks old, the process of oogenesis starts and no more new oogonia is formed and added after birth. Wonderful, right? Let's get to know the stages of oogenesis. The first stage is the multiplication phase. What you see here is the section of ovary with outer germinal layers. Few cells of this germinal epithelium, which are larger than the other cells, functions as germ cells. Can you guess the ploidy of these cells? Yes, it is diploid. These cells undergo repeated mitotic division to form diploid oogonia. In this representation, you can see oogonia protrudes into the cortical region of the ovary as egg tubes. Multicellular mass of these egg tubes form egg nest. One oogonium of the egg nest grows in size and function as primary oocyte, which is still diploid. These cells start dividing meiotically, that is the reduction division, and the first meiotic division, that is the prophase 1 of meiotic division, and gets temporarily arrested at this stage, either diplotin or diakinesis stage. Now, the primary oocyte undergoes the growth phase. Few cells of the germinal epithelium cover it to form the follicular sheath. The end-sheathed primary oocyte is now called primary follicle. About 2 million of these follicles exist in females even before birth. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in the ovary. At puberty, the ovarian follicles start to mature. During this maturation phase, the primary follicles get surrounded by more layers of granulosa cells and a new theca to form the secondary follicle. The cell within is still called the primary oocyte. The primary oocyte within this tertiary follicle grows in size. And now it completes its first meiotic division, producing 2 daughter nuclei, in which the larger haploid cell is called secondary oocyte and the tiny one is called the first polar body. The secondary oocyte retains the bulk nutrient-rich cytoplasm of primary oocyte. Since it has completed the first meiotic division, the chromosome number has got reduced into, now the secondary oocyte is haploid in number. The tertiary follicle further changes into a mature follicle. So this outer covering, which is called as a tertiary follicle, further changes into a mature follicle called Graafian follicle. The secondary oocyte forms a new membrane called zona pellucida around it and starts the second meiotic division, but it remains suspended in metaphase 2. At this stage, the Graafian follicle ruptures to release the secondary oocyte from the ovary. The release of the secondary oocyte from the ovary is called ovulation. The liberated human ovum is alecithal, that is, it lacks yolk. It contains a large amount of cytoplasm, which is otherwise known as ooplasm. Now, outside the ooplasm is a layer of thin membrane called vitelline membrane or the plasma membrane. And this is covered by another layer called zona pellucida. The space between these two membranes, it's a plasma membrane or the vitelline membrane and the zona pellucida, is called as perivitelline space. The final outer layer of the membrane is known as corona radiata, and it is made up of the layers of germinal epithelial cells. We know that the human ovum has just completed the first meiotic division. The completion of this second meiotic division happens only when the sperm enters inside the ovum. So the entry of the sperm induces the second meiotic division of the secondary oocyte, which again divides into the ovum and the polar body. And the sperm fuses, the pronucleus of the sperm fuses with the pronucleus of the ovum, resulting in the formation of diploid zygote. That ends up the process of fertilization.